NBIA, Natural Bioscience Intelligence Agency. The science is real. The mission is yours.
44 states, 18,400 retail locations, 11 SKUs. Every pouch carries a BPA-free claim. Every pouch is lined with BPS. The substitution happened in 2013. It has never been reviewed.
The compound isn't BPA. The regulatory gap was deliberate. BPS, BPF. Structural analogs. Same receptor mechanism. No safety review required. BPA-free on every label. Confirmed present in flexible laminate, across snack pouches, canned goods, and thermal receipts. The system isn't broken. It was designed.
The target demographic, ages 2 through 6. Distribution expanding into 340 pediatric clinic waiting rooms, WIC authorized. The children consuming 2 pouches daily are hitting ER-alpha activation thresholds in pediatric tissue models. The gut lining is still forming. This is a developmental window. They're dosing it.
Similar product categories carry the same threat. Squeeze fruit packs, flexible vegetable pouches, yogurt tubes. Any product in multi-layer laminate with a heat seal adhesive layer. BPA-free is not bisphenol-free. Biochemistry doesn't read marketing copy. The solution is also real and specific. You'll have it before this episode ends. 14 hours earlier, Geneva.
The National Children's Study Archive. 100,000 US children. Urinary bisphenol metabolite measurements. Gastrointestinal health assessments. Birth through age 7. The data has been here for years. BPS and BPF were added to the biomonitoring panel in the study's 3rd year. Before that addition date, the correlation was invisible. After it, the signal appears at month 4. Urinary BPS concentrations mapping against gastrointestinal permeability markers in the 2 to 6 subgroup. A correlation captured in this dataset, never extracted as a standalone analysis. I am the first analyst to align the panel addition date with the GI permeability trajectory data from the same cohort. In children, gastrointestinal epithelial turnover is 3 to 4 times the adult rate. The proliferative signal from ER-alpha activation doesn't produce the same effect it would in a 40-year-old. It produces a faster, amplified version in a 2-year-old. The mucosal barrier is still forming. It is being disrupted during its own construction. The longitudinal consequence of that is not a diagnosis at age 4. It is a chronic condition presenting at age 34, with no paperwork connecting it to a pouch consumed at breakfast when the patient was 2. Joining the mobile briefing from Bethesda. Secure link. Encryption confirmed.
The WIC authorization paperwork for the product SKUs is on screen. Pediatric clinic distribution contracts in the same file. 11 SKUs. 340 pediatric waiting rooms. The formulator acquired the contract manufacturing relationship in 2021. Distribution has expanded every quarter since. Ghost longitudinal summary.
The developmental window for gastrointestinal mucosal maturation closes between ages 5 and 7. What happens to tight junction architecture during that window determines the baseline gut barrier integrity that individual carries for life. The NICHD dataset shows the BPS signal appearing at month 4 of measurability. The permeability markers trend upward. The window doesn't wait.
Before the mechanism a reference point, think of the lining of the gut as a carefully sealed brick wall. Each brick is an epithelial cell. The mortar between them is made of proteins called tight junctions claudin, 4, and occludin. Those proteins are the seal. What BPS does is send a signal that loosens the mortar. The seal becomes permeable. Things that should stay in the gut don't. Here's the mechanism at the molecular level. BPS enters the gastrointestinal tract through food contact migration. The compound moves from the laminate into the food content, particularly under heat or acidic conditions. Once ingested, BPS reaches intestinal epithelial cells and binds estrogen receptor alpha ER-alpha at the ligand binding domain. It binds with sufficient affinity to activate downstream gene transcription. The estrogenic signal it triggers suppresses claudin, 4, and occludin gene expression. The tight junction proteins are not produced at normal levels. The mucosal barrier, the mortar between the bricks, becomes structurally compromised.
If it's suppressing those proteins, what does that actually mean for the gut? What goes wrong?
Increased intestinal permeability. Compounds that should remain in the gut lumen cross the epithelial barrier. Immune activation. Systemic low-grade inflammation. In a child whose gut lining is still forming, the tight junction network is being disrupted during the developmental window when it should be consolidating. In an adult with years of cumulative bisphenol exposure, thermal receipts, canned goods, flexible packaging, the chronic suppression of claudin, 4, and occludin expression is the mechanism underlying what gets diagnosed as IBS, unexplained digestive dysfunction, or unspecified food intolerance. The compound is not considered at any stage of that diagnostic process.
Countermeasure confirmed. Flaxseed linum usitotissimum. Active lignan compound. Sequoia-solariceacinol diglucoside, SDG. The Crop Development Centre here at the University of Saskatchewan developed the high-SDG cultivars CDC Bethune Mons. Canada produces 30 to 40% of global flaxseed supply. The SDG concentration is cultivar dependent and preparation dependent. That distinction is the operational variable.
Explain the preparation variable, Viper.
Whole flaxseed passes through the gastrointestinal tract largely intact. The lignan matrix is encased in the hull. SDG does not reach the colonic microbiome in bioavailable form. Therapeutic SDG delivery requires grinding, freshly ground. The milling exposes the lignan matrix to intestinal bacterial metabolism. That is the difference between a therapeutic dose and a decorative topping. The label has never been required to specify this. Most consumers do not know it.
The longitudinal implication extends beyond the pediatric cohort. Every adult consuming BPS-lined packaging across multiple daily food contacts, snack pouches, canned goods, thermal receipts is accumulating receptor-level estrogenic signaling with no upper age boundary on the mechanism. The tight junction suppression does not stop at age 6. The countermeasure's receptor mechanism is equally applicable across the age range.
Viper. Confirm the compound pathway. All of it.
6 hours later. Crop Development Centre Seed Analysis Laboratory, University of Saskatchewan, Saskatoon, Canada. SDG concentration by cultivar confirmed. CDC Bethune highest lignan density of commercially available varieties. Freshly ground sample prepared. Frost molecular pathway. All of it.
Ground flaxseed delivers SDG to the large intestine intact. SDG is resistant to upper gastrointestinal digestion. It passes through the stomach and small intestine and reaches the colonic microbiome as its primary site of activation. Bacteroides and Clostridium species in the colon cleave the glucoside moiety from SDG, releasing sequoia-solariceacinol SECO. SECO is then metabolized over 24 to 36 hours to enterodiol, and enterodiol converts to enterolactone. Enterolactone and enterodiol are absorbed through the colonic epithelium into portal circulation. Peak plasma concentration occurs 8 to 10 hours post-ingestion. That slow-release pharmacokinetic profile provides sustained receptor occupancy across the day, not a spike, a sustained presence. The BPS exposure through food packaging is also continuous. The countermeasure is designed for continuous deployment.
Steel N3 confirmation on receptor binding affinity differential.
One moment.
Enterolactone and enterodiol bind ER-alpha at the ligand binding domain. Binding affinity is approximately 1/1000th of endogenous estradiol. BPS binding affinity at ER-alpha, approximately 1/500th of estradiol. The lignans are weaker, but they are occupying the same site. Competitive displacement confirmed by receptor occupancy modeling. The weaker signal excludes the stronger synthetic signal when present at sufficient concentration.
Binding displacement confirmed. Frost continue.
In intestinal epithelial cells, enterolactone and enterodiol arrive at ER-alpha's ligand binding domain. They occupy the same site as BPS and BPF. But the downstream signal they trigger is partial agonism, not the full estrogenic activation that synthetic bisphenols produce. Receptor occupancy by the weaker lignan signal competitively excludes the stronger synthetic signal. The BPS cannot bind a receptor that is already occupied. Simultaneously, the NRF2 pathway activates in the same intestinal epithelial cells. NRF2 is the cell's master antioxidant and stress response regulator. SDG metabolites drive NRF2 activation, which pushes claudin, 4, and occludin gene expression upward. The tight junction proteins begin to be produced again. The mortar between the bricks is being replaced. The mucosal barrier does not rebuild in one dose, but it is no longer being actively dismantled.
For a 2-year-old whose gut lining is still forming that distinction, is the difference between a developmental window closing normally or closing compromised. For an adult whose tight junction integrity has been chronically suppressed by years of cumulative bisphenol exposure, it is the difference between a barrier that continues degrading and one that begins to recover. The mechanism does not check age at the door, neither does the damage.
So the flaxseed isn't removing the BPS, it's just getting to the receptor first?
Competitive displacement at the receptor level. The SDG metabolites do not neutralize BPS as a compound they occupy its binding site. The BPS is present. The receptor is unavailable. The downstream estrogenic signal, the one that suppresses claudin, 4 and occludin, is reduced. That is the mechanism. The formulator's operation stalls at the receptor. Not upstream, not downstream. At the entry point of the disruption.
SDG delivery confirmed. CDC Bethune cultivar, freshly ground. Mission package complete.
The formulator's pediatric pouch distribution is documented. WIC authorization file transmitted to NBIA legal division. Pediatric clinic contracts flagged for institutional review. The compound is in the supply chain. The regulatory gap that permitted the substitution has not been closed. Our countermeasure is confirmed and sourced. The mission is the consumer's awareness, not their ignorance.
The niche decorrelation data is being compiled as an extractable sub-analysis for the first time. The signal was always in the archive. It required the right question. The right question took 4 years.
Saskatoon is clear. Returning via Sacramento route. Pouch samples secured for independent analysis.
Sodium channel modulation confirmed at 15 parts per million. Neuronal firing threshold reduced by 23% at standard dietary exposure frequency. The behavioral presentation is indistinguishable from attention deficit in the 6 to 12 demographic. Distributing through the school breakfast program, the volume numbers are exceptional.
Paper trail is complete. BPS displacement mechanism confirmed. Countermeasure documented and sourced. GHOST, the NICHD sub-analysis, goes to the Scientific Review Board this week. Frost archived the Stinger citation set. Viper debriefing in 48 hours.
The school breakfast program has distribution in 53,000 facilities across 31 states. If the sodium channel modulation signal is confirmed at dietary exposure, the behavioral presentation window for that cohort is every morning, repeated daily. The volume numbers he referenced are not exceptional. They are unprecedented.
Operation. Frequency is authorized. Next operation begins now.
Everything you're about to hear has been published in peer-reviewed research. I'm going to give you the mechanism, the study, and the exact dose in plain language. The mechanism. Flaxseed contains a plant lignan called sequisolarisiercinol diglucoside SDG. When you consume freshly ground flaxseed, SDG reaches your large intestine intact and is metabolized by your gut bacteria into 2 compounds, enterolactone and enterodiol. These are phytoestrogens. They bind the same estrogen receptor that BPS and BPF bind. Because they occupy that receptor site, they reduce the estrogenic signal that synthetic bisphenols produce. The downstream result, the genes that produce your gut lining's tight junction proteins, claudin, 4, and occludin, are no longer being suppressed. At the same time, SDG metabolites activate a cellular defense pathway called NRF2, which actively drives those tight junction proteins back upward. The damage mechanism is interrupted. The barrier begins to recover. The study. Sprague and colleagues, Environmental Health Perspectives, 2014, volume 122. Human epidemiological analysis of children aged 6 to 11. Urinary BPA concentrations were significantly associated with increased gastrointestinal inflammation markers and reduced tight junction protein expression, consistent with ER-alpha-mediated epithelial disruption. A supporting mechanistic study by Shafrir and colleagues, Food and Chemical Toxicology, 2010, Volume 48, confirmed that SDG metabolites enterolactone and enterodiol competitively displaced BPA at ER-alpha binding sites in intestinal epithelial cell models. The dose: 1 to 2 tablespoons of freshly ground flaxseed daily. That provides approximately 50 to 150 milligrams of SDG, depending on cultivar. The detail the industry does not put on the label: whole flaxseed delivers negligible SDG bioavailability. The hull encases the lignan matrix. It passes through your gut intact. Only ground flaxseed freshly milled exposes SDG to the bacterial metabolism that converts it to enterolactone and enterodiol. Buy it whole, grind it fresh. And understand this: BPA-free packaging is not bisphenol-free packaging. BPS and BPF are structural analogs with documented ER-alpha activity. The regulatory gap that allowed their substitution without safety review has not been closed. NBIA, Truth and Transparency in Human Health.